Good afternoon, dear students. Today we are going to discuss about Vdrl test. What does Vdrl stand for? Vdrl stands for Vdrl Disease Research Laboratory. This test is done for the detection of a disease called as syphilis. Since the test that is required for detection of syphilis was first carried out in the Vdrl Disease Research Lab, hence the name of the test is given as Vdrl test or Vdrl Disease Research Laboratory test. This is actually a non-specific test used for the rapid screening of syphilis. Okay. So, Vdrl is a non-specific test, that means non-treponemal microflocculation test used for detection of syphilis for rapid screening of syphilis. In this test, we detect IgM or IgG antibodies that are formed against lipoidal material. That means the test detects anti-lipoidal antibodies which are not real antibodies and hence they are called as reagin antibodies. Okay. So, in Vdrl test, we actually detect anti-lipoidal reagin antibodies released from the damaged host cells. Okay. As well as antibodies that are formed against lipoprotein-like material, possibly cardiolipin released from the treponemes, right? So, Vdrl test is a non-specific, non-treponemal test used for rapid screening of syphilis. The test detects antibodies formed against, or we detect anti-lipoidal reagin antibodies. Okay. Now, what is syphilis? Syphilis is actually a sexually transmitted disease, also called as venereal disease, which is caused by spiral-shaped bacteria, that is spirochete, named as Treponema pallidum. So, what is the causative agent of syphilis? The causative agent of syphilis is Treponema pallidum. The organism cannot be cultured in artificial media and hence diagnosis of syphilis depends upon the correlation of the clinical data. When the patient goes to the clinician looking at the symptoms, so we correlate the clinical data with the demonstration of microorganism in the lesion or by serological testing. Okay, so what is the causative agent of syphilis? Treponema pallidum. Treponema pallidum is a spirochete, that means it's a spiral shaped bacteria. You can see here spiral shaped bacteria. This is as seen in dark field microscopy. Okay, spirals. This is again another method of staining where you can see spiral shaped bacteria. So this is the slide. These are the slides that show spiral shaped Treponema pallidum. Now, how the disease is transmitted? As I told you, it's a venereal disease. It is a sexually transmitted disease and hence transmitted through sexual contact, or it can be transmitted by vertical transmission, that is from mother to fetus or from infected blood also. Okay, so now the disease is transmitted through sexual contact or from mother to fetus or from infected blood. Now, how to detect the disease? There are two different types of tests that are used for the detection of the disease. That is specific test, wherein we use treponemal antigen. The causative agent of the disease is Treponema pallidum. Okay, so the serological tests are divided into two categories. Number one, specific test, and number two, non-specific test. In case of non-specific test, we use non-treponemal antigen, wherein we are not going to use the antigen of the disease causing organism. We use non-treponemal cardiolipin antigen. Okay, and what are the different non-specific tests? We can you carry out simple traditional Vdrl test, or nowadays we have RPR test, that is rapid plasma reagin test, or we can use trust antigen test, toluidine red unheated serum test. So these are all the non-treponemal, non-specific test where we do not use treponemal antigen. Then what we use? We use cardiolipin antigen. In case of traditional VDRL test, we use cardiolipin antigen, but we have to heat the serum. But there is slight modification in VDRL test. Nowadays we have modified VDRL test like RPR test, rapid plasma reagent test, again a non-specific non-treponemal serological test, but here the cardiolipin antigen are coated with carbon particles, charcoal particles. So the reaction can be seen as black floccules. Okay. In case of trust antigen test, the long form is toluidine red unheated serum test. In case of trust antigen test, again the cardiolipin antigen, non-specific non-treponemal antigen is coated with a red dye, that is toluidine red, and hence the antigen is red in color, and the flocculation reaction will become more easily visible because of the red colored pigment that is attached to the cardiolipin antigen. So the cardiolipin antigen is actually coated with a red dye, toluidine red, so it appears red in color, and here we don't have to heat the serum. It's a modified VDRL test. And okay, and today in our laboratory also we use trust antigen test for detection of syphilis. Now, once you get this non-specific serological test positive, that is if you are performing trust antigen test or RPR test or normal traditional VDRL test, and if it comes out to be positive, then that means the serum is reactive. Then we have to confirm the results by performing specific test. Specific tests are those where we use treponemal antigens, specific test, like fluorescent antibody testing, okay, then treponema pallidum heme agglutination test, which is called as TPHA test, okay. So fluorescent antibody testing, TPHA testing, where we use treponemal antigen, those tests are used to confirm the results that are obtained by VDRL test, okay. Now, so serological procedures used for detection of syphilis. They use specific test. What are those? Treponemal test. But specific tests are used only after we get non-specific test positive, right? So, they are treponemal test. For example, they detect the antibodies formed against treponema pallidum. They detect antibodies formed against treponema pallidum. That means, okay, and hence we use treponema antigen here. For example, fluorescent treponema pallidum antibody absorption, FTA-ABS in short we call it. Fluorescent treponema pallidum antibody absorption test. Then micro heme agglutination test, MHATP, okay, or TPHA, treponema pallidum heme agglutination test. All these are the specific tests. They are used to confirm the disease. Okay, and the first test that we perform are the non-treponemal test or non-specific test, which and what does these tests, non-specific tests, detect? Reagent antibody. They detect reagent antibodies, antibodies which are anti-lipoidal. See, whenever the infection occurs in a patient, then the cells are damaged. The damaged cells release lipid molecules, lipoidal molecules. Against these lipid also antibodies are produced in the blood. So in non-specific test we actually detect antibodies formed against the lipid molecules released by the damaged host cells, right? So non-treponemal non-specific tests detect the antibodies produced in response to the lipoidal material. That means they are anti-lipoidal antibodies released against lipoidal material released from the damaged host cells. These antibodies are traditionally called as reagent antibodies. Okay, so VDRL test is a non-treponemal, non-specific test. RPR, rapid plasma reagent test, trust antigen test, all are non-treponemal, non-specific test. Now let us understand. So these are the various, we can say, symptoms that appear during syphilis. Is actually divided into 3 stages. Primary stage, where the infection has occurred, and in few weeks to few, you can say, years after a period of 6 or 9 months, primary symptoms appear, and cankers, or rather you can say some kind of lesions are seen in the mouth, oral or genital parts and all. right? And in the second stage, rashes are seen, and in the final stage the disease may reach the brain, right? So there are various symptoms. So lesion or canker actually appears on various body parts during the initial stages of syphilis, and in the final stages the whole body may get covered with the lesions or canker, which contains some kind of fluid, and the fluid is also responsible for causing infection. Okay. Now, what is the principle of the test that we are using? What we are detecting, once again, we are using non-specific test. So, we are going to detect antibodies formed against lipoidal material released from cells damaged by treponema bacteria. So, we are detecting antilipoidal antibodies, which are called as reagent antibodies. And what is the antigen that you add? Antigen is actually a cardiolipin antigen. Okay. Now, the presence of lipoidal antibodies in patient serum or in the serum or CSF. In the final stages, the disease reaches the brain. So, CSF may also be used as a sample. So, we detect lipoidal antibodies, antilipoidal antibodies in patient serum or CSF by using non-specific antigen that is suspended in buffered solution. What is it? Cardiolipin antigen. So, actually, it's a alcoholic solution which contains cardiolipin, lecithin and cholesterol. Now, we sometimes in traditional test we use heated serum, but in nowadays in RPR test or trust antigen test we don't heat the serum. So, the inactivated serum or CSF is mixed with the cardiolipin antigen. Okay, if the patient's serum contains antilipoidal antibodies, it will react with the cardiolipin antigen, and the antigen antibody reaction can be seen as floccules, that is flocculation reaction. To make the reaction more visible to the naked eyes, nowadays the cardiolipin antigen is coated with toluidine red dye or it is tagged with charcoal particles to make the reaction visible, right? Now, so if the patient serum is reactive and contain antilipoidal antibodies, then you can see flocculation. In that case, the serum is said to be reactive. If the patient is negative for syphilis, then the serum will be non-reactive and we won't be able to see any antigen antibody reaction. That means no flocculation will occur. So, non-reactive specimen appears as homogeneous suspension, whereas reactive sample give flocculation reaction. So, we have to check the reaction at the end of 8 minutes, right? So, we have to bring the Vdrl antigen suspension control sample to room temperature, and then 50 microliter or 1 drop of test specimen is taken in the first circle of the, you can say, reaction card or a slide. In the second drop, we take positive control serum, and in the third drop, negative control serum is taken. And then we mix 1 drop or 50 microliter of cardiolipin antigen in with each of the sample, that is with the patient sample with the positive control and negative control. Mix it with the mixing stick properly and then spread it uniformly in the entire reaction circle. Rotate the slide gently, either mechanically or on a rotor at 180 RPM, and observe the flocculation reaction after 8 minutes. If the serum is reactive, you can see here dark carbon particles, black colored particles, the serum is reactive. And if it is weakly reactive, you may not be able to see much prominent black colored floccules. Okay, and in case of non-reactive, you get a homogeneous mixture. So at the end of 8 minutes, you have to report whether the serum is reactive or non-reactive. If the serum is reactive, we say that Vdrl test is positive and it has to be confirmed by a specific test. Okay, this is rapid plasma reagent test. You can see black colored floccules here because the cardiolipin antigen is coated with charcoal particles. So the reaction is more visible. The flocculation reaction becomes more easy to visualize. Now, the next one that we use generally in our laboratory is trust antigen test. Again, it's a modified Vdrl test. Okay, it's a macroscopic non-treponemal flocculation test. Right? In trust antigen, the test particles of so the particles of toluidine red are used in place of charcoal, which is used in RPR. You can use serum or plasma, and it helps you in, you can say, it improves the visibility. And okay, toluidine red, which is tagged to the cardiolipin antigen, will improve the visibility of the floccules that are formed. It is just a VDRL test but modified. Here you don't have to use the heated serum. Okay, so that is all about VDRL test, which is used to detect syphilis. It's a screening test, a rapid screening test for detection of syphilis. It's a non-specific test because we use non-tryponymal cardiolipin antigen. Okay.